Yeah, at the place of Eduardo should be present here, but he had a last-minute issue, so he sent me to present this place. And actually, following the line of the previous presentation, in this case, we also apply a host diet therapy to promote the clearance of Mycobacterium abscessus infection by trying to promote the ability of the macrophage to kill the bacteria. So I will go really fast on the next slides, since we already got to know Mycobacterium abscessus, but it's like, as we got to know, it's an opportunistic pathogen, so it's widely present in environment, and mainly, in this case, infect people that are immunocompromised. Whenever then we got an infection, what happened that alveolar macrophages, which has residence macrophage in the lung, quickly recognize the pathogen and internalize it, and then they have the recruitment of the other immune cells and the formation of the granuloma. And we decide actually to adhere, because what the bacteria does is to try to actually compromise the ability of the macrophage to clear the infection by promoting what is the anti-inflammatory response. So, as I told you before, the main macrophage that intervene at the first step of the Mycobacterium abscessus infection are alveolar macrophages, which are a bit different from other like circulating macrophage, because first of all, they're not derived from the bone marrow, but they're derived from the yolk sac. And differently from other like differentiated macrophages, they are able to proliferate in situ. Their proliferation is not amazing, they just like there is 10–20%, but they're still able to proliferate to replace the macrophage that get activated, they die. And because of the lung environment, which is a direct contact with external environment, they have to be able to kind of tolerate all the pollution or the particles that come from the external environment, and therefore they have high plasticity in order to respond to the better way possible to this external stimuli. So, as I told you before, when we have the infection, the macrophage internalize the pathogen, and we have the activation of the pro-inflammatory response, where they try to clear the infection. But the bacteria is quite clever, so what it does, it tries to promote what is the alternative activation, the macrophages, in order to allow its survival and proliferation. And we ask which are the mechanisms that actually intervene in this process. And among the many mechanisms that intervene, we focus on what is the epigenetics of the macrophage and their activation. Why epigenetic? Because they alter the genetic code. They are really fast, and they can change rapidly depending on the stimuli we have. They provide an extra layer of transcriptional flexibility, because they're really happening really fast, and depending on what the stimuli is, they can change in a time-dependent manner, and also they depend on the activity of two enzymes, which are the writer, they add this epigenetic modification, or erase or they remove. Among the many modifications, we decide to focus on the acetylation of lysine 14 or the histone 3, and this is because actually we know that in published clinical data it has been shown that people that have active and severe form of tuberculosis have actually an hypoacetylation of this lysine. And also has been shown that this modification associated with M1 phenotype in macrophages, and also increases gene transcription of some genes intervening in the inflammatory response. So what we did as first was actually to check in an in vitro system that closely replicate alveolar macrophages mouse if we have changes in acetylation of our macrophages after Mycobacterium abscessus infection. And here you can see the bacteria in yellow. And what we actually saw, with respect to the control, the cell infected actually a lower level of acetylation, meaning that actually the pathogen is doing something. And it also was more interesting that if we discriminate the cell that actually internalized the pathogen, the level of acetylation of this lysine 14 was even lower with respect to the cells that only sense the pathogen. So at this point, if we know that when the pathogen is internalized, we have a reduction in this acetylation, we wonder whether we can use some molecules that can pharmacologically reverse this modification. In particular, we decided to use already FDA-approved epigenetic modulators in order kind of to accelerate the use in clinics settings. Among the many modulators that we find out, we focus on panobinostat, which is actually well known and well characterized, is a non-selective histone acetylase inhibitor, so it acts on several diverse acetylases, and at the moment is used as a treatment of multiple myeloma. In general, is used to lead to increase the apoptosis of malignant cells, but our idea was to use it as subclinical concentration in order to avoid the cytotoxic effect, and only to use which is these epigenetic modulators. So as a first things, we want to check if using our in vitro system, clinical concentration of panobinostat could induce apoptosis in our cells, reduce cell viability. And what we saw that if we use 6 nanomolars of panobinostat, we actually can keep cells quite viable up to 48 hours. Then what we checked was if the concentration that we used was actually able to induce the increase in the acetylation level of the lysine 14. This is some confocal macroscopy that we carry out in our cells. And actually what we saw that the lowest concentration was able to induce the highest actually increase in the level of acetylation, especially at the first hours, which for us was not surprising because if we think epigenetic modification happened really fast. At this point, what we did was to characterize a bit better which were the effect of panobinostat in our cells, and we carry out whole cells proteomics thanks to our collaboration with Professor Altomare at the University of Milan. And this is like this is a volcano plot of the cells of our proteins. And actually, what was surprising is that the concentration that we use, which was 6 nanomolar, so really low respect to the concentration normally using, was reducing all the apoptosis pathway and promoted cell viability pathway. Moreover, was increasing autophagy, lysosomal activity, which may be important both in the recognition of the pathogen, but also in its clearance. And this was most surprising. So it was a reduction in lipid metabolism and increase in the carbohydrate one. And this was interesting because when we know that when the pathogen wants to promote the anti-inflammatory response for the macrophages, it induces a switching in metabolism, increasing which is the use of lipids while reducing the use of carbohydrate. Then at this point, we decided to check what happened during infection. So we used as a system, we infected with our pathogen for 1 hour, and then we treated ourselves with panobinostat for 1, 3, and 6 hours, and we check at the transcriptomic level what happened. We check a bunch of anti-inflammatory genes, and what was surprising is that while the infected cells, but not treated, showed an increase in all the anti-inflammatory marker, which might suggest that actually pathogen is promoting the dampening of the infection, panobinostat was either able to decrease it or to keep it stable, suggesting that actually the panobinostat can help the cells to fight back the mycobacteria. As a next step, we also check PPAR gamma, which is important in normal state to keep the macrophage actually in their non-activate state, and also in this case, we have a downregulation in cells infected and treated, and this, we check CD36, which we know they promote the lipid metabolism switch, and actually in this case, we found the panobinostat to keep it actually downregulated with respect to infected cells, meaning that actually panobinostat is really helping the cells to fight the infection. When we checked then pro-inflammatory genes, actually we were a bit surprised because we didn't observe any actually upregulation in the gene in cells that are infected and treated. But then actually thinking about that, we were not surprising because we know that if we increase too much, which is the pro-inflammatory gene, it can be detrimental for the infection and this promoted in dissemination. As a last thing, we actually check if panobinostat was really decreasing the intracellular viability of the bacteria. So we have our cells, we infected and treated the cells, we lysate and carry out some CFU counting to see MAB intracellular viability. And what we we're happy to see the panobinostat was able to reduce MAB intracellular viability, especially the first time point. So as conclusion, we know that also MAB can actually induce the regulation of the acetylation level of the lysine 14 and histone 3. Well, we know that if we use then panobinostat as subclinical concentration, we can kind of regulate back the level of acetylation to lysine 14, but we don't have any acetylation, any cytotoxic effect, and that it might help the cells to kind of fight back or to avoid the hijacking of the MAB, and also that the cells that were treated with panobinostat can actually reduce the pathogen intracellular viability. As a next step, we actually would like to check if panobinostat has the same effect in longer, like in prolonged time, check if we can have synergic activity with other antibiotics, as ciprofloxacin and amikacin, and in collaboration with the unit of Professor Cirillo, we like to check if the panobinostat has an effect in vivo. With this, I'd like to thank the lab that was actually yesterday. In particular, I would like to thank Stefano, which did most of the bench work, our collaborator at the University of Milan, Alessandro Artomane, Professor Aldini, also our collaborator and partner, Raffaele, Professor Daniele Cirillo, Nicola, Fabio, Federica. We're actually carrying out the work in vivo. The delegation that actually adopted our project, and the foundation, of course, and you for your attention.